Welcome everybody. We're starting. We're very happy to have this meeting in conjunction with the French community. We're very glad that the same event that we hold monthly here, so we have talks in multidisciplinary fields every month. And we gather the Brazilian community and we talk about research and innovation in Portuguese. And the French community, French-speaking community does the same. And we decided to get together and make this meeting with both communities. I want to thank Joelle for making this connection. And I want to also thank the Minister Roberto Medeiros, who is representing the Consulate General of Brazil in Boston. I want to thank Cyber Foundation, who is sponsoring the space for this event as well.
So I'm Valérie Philippon. I represent the scientific cafe, the French scientific cafe. So it's really the Francophone. So we're actually a group of 7 Francophile and Francophone groups in Boston. We have the French consulate in Boston. We have Swissness, representing the Swiss community. Wallonie, Bruxelles for the Belgium. Also working with the delegation of the Québécois in Boston, in Canada, as well as the Franco-American Chamber of Commerce, French Day, and the representative from the CCE, which is the French Trade Advisor, who also have a group in Boston. So there are 7 different organizations of French-speaking folks in the community, and we've been meeting on a roughly monthly through the past 10 years to have Scientific Cafe in French. So we're really excited to have this joint meeting with the Brazilian community today. So it's a great opportunity to join our scientific mind. So with that, we have a French and a Brazilian speaker tonight. We're going to talk about mosquito-borne illnesses. So let me first introduce Millicent. So Millicent is an instructor at Harvard Medical School. There we go, much better. And our current research interest involved, you can see the development of squad inhibitor for fevereiro. So she received her PhD from France at INRA Versailles, and then she moved to the US to do her postdoctoral work at the University of California, Riverside, when she studied RNA replication and encapsulation of viruses from the Bromoviridae family. So for a few years, from 2008 till 2011, Millicent actually worked in Lucy Yang's laboratory at Harvard Medical School, then went back to France, and now she's back in the US. So she really has the experience from both sides of the ocean on scientific development and work on these viruses. So thank you, Millicent, for having accepted our invitation. And I'm looking back on this one.
So thank you all for the invitation, and I will talk today about the identification of slow-molecular viruses. And so viruses are arboviruses, which means they are arthropod-borne viruses. So it means that the virus is originally amplified in insect vectors like mosquito or ticks, and you have some amplification cycles that involve like vertebrate species, which means like through a bite of a vertebrate species, you have like the virus can multiply the other host, such as pigs or human. So what is interesting to us is the human host. So for instance, in the case of dengue virus, for instance, or chikungunya, the human host sustain a very high uremia in blood, which makes it able to go through another amplification cycle when a mosquito or a tick is fighting the human. Then you have, for arboviruses, you can have also different type of cycles that involve like that, what we call dead and host, which means that the host, such as human, for instance, is not able to sustain a high uremia in blood, which means that the mosquito is going to fight the human again. And that's the case of some viruses that are tropic viruses. And so today I'm going to talk about some several flaviviruses. I will mostly talk about Japanese encephalitis virus, which is a mosquito-borne virus transmitted by Culex species of mosquitoes, and which has amplified host is pigs and developed generally neuropathic disease in human. And I will talk also a lot about dengue virus as well as Zika virus, which are both involved in primate cycle and involve mosquito vector. So, yes, so you don't see anything, but this is supposed to be a map of the last month cases of all those of a bunch of human anyway. This is not a high season for mosquito transmission, which is why it's not as many cases as we would have in different time. But like what is important to note is like there are some very effective vaccines against yellow fever and Japanese encephalitis that have been available since the 30s, but there are still some cases of this disease around the world. And the viruses I'm going to talk about are Japanese encephalitis virus, which is restricted to Southeast Asia. And there's several vaccines that have been available. There's no antiviral, but it still remains to be a high public health threat because there are many people circulating in this region, and there are people that are at risk. And it is a neurotropic virus. So because of the lack of antivirals, you have, anyway, it's a public health problem. You've all heard about Zika virus. I'm going to talk about Zika virus, for which there's no vaccine and no antivirals. Of course, the most important virus is dengue virus. It's thought to be one of the most important mosquito-borne illnesses. It has 3.9 billion people at risk in like, a lot of countries around the world. And there are several vaccines in development, which vaccine that was developed by Sanofi in 2016 does not confirm protection for people that have never been exposed to the vaccine. And there's no antiviral against this virus. So, just to give you a little background of flaviviruses, it's a single strand positive stance RNA virus, which means that the viral genome is a messenger RNA that is going to be translated to polyprotein that is processed by host and viral proteins into 3 structural proteins and 7 non-structural proteins, and the variant is an enveloped chemical variant. The infectious cycle starts by the virus attaching to the cells through an unknown receptor. There is upon low pH in the endosome, there is a virus fusion occur and to release of the genomic RNA that is then translated, and viral RNA replication happens in the ER membrane. Then the virus is building through the secretory pathway and secreting the cells. So in Priscilla Young's laboratory, we are interested in the development of small molecule antivirals, and there are like many campaigns of screening campaigns that led to the integration of small molecule inhibitor of dengue virus, because that's a virus we are mainly interested in. And when we identify a small molecule antiviral, we ask two questions, which are what is the target of the small molecule inhibitor, and where is the block happening in the viral infection? So this is a question we have answered for several of the inhibitors we have identified, which might be like the directly eating the virion, okay, some host factor. And this is useful for basic biology because those small molecule inhibitors, once we know how they act, they can be used as a probe to study some aspect of basic biology. And also, it's interesting when we identify new antiviral targets, when we want to for antiviral discovery and development. So I will talk today about two aspects of my work, which are one, the inhibition of flavivirus by targeting a viral protein, and the second part will be about targeting host factor involved in infection. So I will talk about inhibitors that are targeting this early stage of infection, which is entry. You can imagine that if you block this stage of infection, you are successful at eliminating the infection of the cell, so it's a very attractive target. And to go through the way, this is just like showing you how fusion happens. So fusion is this step when the virus is using its membrane with the host cell membrane and leads to the basic phase of the genomic RNA in the endosome. And this is the envelope protein, which is present at the variant surface as a dimer. And this is a color code for the viral protein. And what's happening is like due to a series of structural rearrangements, we're going to have insertion of the fusion nucleus inside the host lipid membrane, and you're going to have fusion happening, and which is going to bring the viral and the host membrane together, which leads to the release of the genome. And so through phenotypic screening, previous members in the lab have identified three classes of small molecule that are targeting dengue virus envelope proteins, so that directly bind to this protein, and that they act at inhibiting entry process by inhibiting viral fusion. So this is very process. And so to take you back to earlier work done by when he crystallized dengue virus envelope protein, what he found is that this detergent molecule called beta-octylglucoside bug was co-crystallizing in this pockets between the domain 1 and domain 2. And so the thought at the time was that like maybe because there are so many structural change happening to fusion, if you like are using this little pocket and you can target it with small molecule inhibitors, you can make it fusion. And so the current hypothesis in the lab is that like those small molecule inhibitors that I just showed are inhibiting fusion of antivirus are acting in the same way, in that way, and they are targeting the bug pocket. This is just a talking of one of the small molecules in this pocket in the antivirus and bug pocket. And so to one question that I was interested in is because this bug pocket is actually concerned with flavivirus envelope protein. This is a structural alignment of dengue with Japanese encephalitis virus and Zika virus. And in the red is the bug pocket. You can see this is structurally conserved. And the question was like, can we target this pocket for, because this is conserved, maybe our small molecule would exhibit antiviral activity against these other viruses. So this is what I, just to take you back on how do we quantify antiviral activity, because we are looking at inhibitors that are targeting the virus. So when we do this is we incubate the small molecule with the virus for 45 minutes, 37 degrees, so that the small molecule has a potential to enter the virus, and then we perform the infection of cells for 1 hour, which is the time that is needed for the virus to enter the cell and fuse, or not fuse with the small molecule this event. And then we wash out the inhibitor, we increase the cell for 24 hours, and then we are going to collect the supernatant that are containing the infectious virus. And we quantify the amount of infectious virus in the supernatant by standard conformal increasing. And because we incubate with range of concentration of small molecule, we can determine what we call the IC90, which is a way to quantify how potent the compound is. And it tells you this value is showing you what is the concentration of compound is needed to reduce the yield of infectious virus by 10. And that's what I'm going to mention when I talk about the potency of this compound. And so when I try to, so those compounds have been, these three classes of compounds have been shown to be active against dengue, and they have different EC90 against dengue virus. In this assay I just described, with the more potent is the cyanohydrazone. When I tested the antiviral activity against Zika virus, they were conserving some antiviral activity. The cyanohydrazone through ATM22 have like further antiviral activity with value close to that of against dengue virus. And when I tested them against, we tested them, so some students worked with this assay against Japanese encephalitis virus. We lost some of the potency, which kind of makes sense because Zika virus is actually closely related, more closely related to dengue than GBV. So we validated this pump-caliber's antiviral activity, and we think that the both pocket can be like a good target for antiviral. So now we switch to the targeting host factor with small molecule. And why do we think targeting host factor is important, is interesting? Because like many viruses actually uses the same pathway to infect cells. So if you target the host factor, it might have potential for the antiviral activity against other viruses. And another advantage is there is less risk that the virus is going to evolve resistance. When you target a viral factor, you can evolve rapidly to be resistant to the small molecule. And so the strategy we took to develop those targeted antiviral is to use the covalent inhibitor. Covalent inhibitor, what it means is that there are inhibitors that are forming a covalent bond with their target protein. And it's a nice thing to have because it has much higher potency than compounds that are not this covalent bond. And when you treat them, you can expose the cells for much less time to have like potency. So it does prolong pharmacodynamics. And also you can utilize this for targeted identification. You can convert to small molecule protein complex and identify what protein is targeted by the small molecule. So it's very interesting antiviral discovery. And currently, that's a very valid strategy for drug development. Many drugs are the DAs so we did a collaboration with Nathaniel Gorilla at the Nathaniel Institute, and we screen libraries that you have available that are heterocycle acrylamide libraries. So the acrylamide, as I say, is the one that is the warhead that is forming covalent bond with cysteine. Cysteine are very important active residue proteins that correlate with the activity of the protein. And there's another chemistry part of the small molecule that is targeting ATP sites. And interesting fact about this library is that they know already what kinase those small molecules are targeting. They have a very good idea of what we are looking at. Small molecules we did 2 screens. The screen I was involved with involved the use of proton viral particle, and I will describe what those are, but like briefly we are interrogating early stage of viral infection from entry to RNA activation. And Michael Peter, a former postdoc in the lab, looked at the screen using a replicant system, which is a way of translation and information. And so what are proton viral particles? I will just explain. So it uses a replicon. A replicon is a flavivirus full-length RNA, but you deleted the structural protein, which means this RNA is not able to form an infectious virus because it does not have the structural protein. But it's able to replicate, and you can replace the structural protein with whatever protein you want. And when you co-express in the cell the structural protein, you are making viral particles that encapsulate this replicon. So you can infect cells in less biosafety setting and look at very rapidly at the vanilla secret signal, important signal as a measure of infectivity. So that's the screen I made, and my colleague did another screen where I use stable cells which are only the replicon in the cell, and you look at inhibition of this replicon, which they do by the and so the small molecule we identify in this screen were developed the most potent small molecule we identify in both of the screen belong to the QL compounds named after Chinsamlu with the postdoc in Maternal Lab and made those compounds and designed them to target the BTX-DMX kinases. And the setting of the antiviral activity is a little different because we infect the compound post-infection because those are targeting this factor instead of targeting the virus. But they have like very potent activity against antivirals and infectious assays. It's actually quite passive. You are talking to this is star compound. We just said activity. And we use those who are targeting these kinase inhibitors, but when we use other compounds that are not to target these kinases, we found that there's no correlation with antiviral activity. They do not exhibit antiviral activity, so those are not the target of the compound, and we are still doing studies to identify what is the target of those compounds. An interesting fact is that when we remove the acrylamide, which is the one that is forming covalent bonds to the protein, we lose activity. And also because those are covalent inhibitors, you can just treat the cells and then infect the virus, and the virus is no longer able to infect the cells because you have this covalent inhibition that is made. We just show you how this is going on. And so, as I said earlier, when you find me, the question we ask is how, what is the mode of action, what step of infection is affected, and we're looking at something the early event of infection, translation or activation. So we look at that and we use this RNA replicon that I just described, which shows that as expected, the compounds are inhibiting RNA replication. If we mutate the polymerase in the replicon, here, we're only looking at translation of the reportogen, and with that we found that the compound are in translation of the reportogen. So what we found is that those compounds are actually in viral protein expression and accumulation. So we think that's the way those compounds are acting. And it correlates with antibiotic activity. And then Yankee worked a lot with microbiology also in the lab, and they did a lot of other compounds of the same derived from this family and did a lot of medication. And what they saw is like when you have compounds that have higher antiviral activities, you have a higher inhibition of translation, and you can use that. So it's a very good correlation between antiviral activity and inhibition of viral translation. And that actually holds true. What I started saying is that the interest in this type of molecule that target host factor is that it can be whole spectrum antiviral, and it is actually true because we tested against other flaviviruses like Aspergillus, Zika virus, which are other flavivirus, and they were inhibited by our star compound, but also we did some other testing, mostly we did some collaboration with Aiden and Shelley. We tested some other pathogen and we also tested other viruses we have in house, and it inhibits very wide a lot of other viruses that are not flaviviride, and I could show that it actually was inhibiting protein expression for many of these viruses. And so just to conclude, I just showed you that we are developing the envelope protein of the virus, and those inhibitors can inhibit other flaviviruses. Our ongoing effort on this aspect of the project is to check if the mode of action of this small molecule is the same as against dengue, which means are they actually also inhibited fusion, and to validate the interaction with the concept of focus. And we also identify Delta 47 as a host targeting inhibitor that's by extension, and now ongoing for how to more characterize the mode of action of this molecule, how specifically does this compound expression, and we are working a lot on target and lighting inhibition, which is going to help us determine how this compound works. We also have like better compounds like better which derive from this consciousness of the percent and finally, I would like to thank for Priscilla, who's my mentor, that's supportive of me. My labmates, especially so Patrice, since again she's student involved now on the work, Isabella was a student working on this. Marco was working with us a lot, and we have a good collaboration in that.
I work on the parasites and it's responsible for malaria. Engineer to resistance. That compound can you do the same with virus?
Yeah, so it's like more like so for the entry, it's actually something quantified. It's actually very interesting. So for the entry, because actually the resistant viruses that are mapping. So this is another way to validate this property. For QM247, because it's a host study, I try to resistant viruses. it's really, so it's like, and I think it's actually multi-targeted. So.
I have two questions. The first is when you're talking that they inhibit different viruses, how different is the dose? Do you have to go for micromolar, nanomolar to micromolar?
Also, actually,
Yeah. For the second one,
the second one, no.
is it constant value?
Yeah, no, it's those experiments are done all at 2 micromolar. And it's kind of difficult to evaluate because this virus contributes much higher titer. So comparing them amongst viruses, but in general, 2 micromolar.
And when you say the second one is when you say you want to test in vivo, being a close target that you don't know, do you think you're going to have to find an equivalent inhibitor for mice, or which model?
Yeah, we're using mice. So before doing vivo work, of course, we validate that on mouse it is effective. The mouse strain for mice. But yeah. So it is.
Yeah.
First of all, thanks for the presentation. So just to make sure that I understood. The first part, the inhibitor of the fusion, you did these studies for showing a reduction of the fevereiro, right? In the first part,
Yeah.
you did the studies to show reduction of the fevereiro.
Yeah.
And you got to the conclusion that the mechanism of action is inhibition of fusion based on molecular modeling. Do you have any biological result that?
No, so I didn't show for dengue virus. We actually got results that is going to be published soon, but we have a lot of characterization of the mode of action showing that it's actually biochemical characterization. And as she mentioned, like using resistant viruses, and you may take like cycle interactions, small molecules. So for dengue, we actually showed very convincingly that it happens through that mechanism. Now for the other viruses, we are lacking data, and that's why it's really important for us to show that it's the same.
Well, I have two questions, but I'm going to ask them in the dengue virus, I thought it was a disease a bit like a flu that was not really life-threatening except for young kids. So, what's the purpose of the drug? How do you think that a drug like that would be useful?
So it's actually life-threatening for people that are exposed to infections. There's this thing called antibody. So it is bad for kids. So, but like you have like 4 serotypes of dengue that are circulating that are like unrelated enough that like you can develop antibody against one serotype, and then this is going to help another serotype to infect cells instead of like blocking the other serotype. And that's the problem with the vaccine. That's why vaccines are not recommended for people who have never been exposed, because the vaccine is going to act the same way. If you never been exposed to dengue, it's going to do dengue. I'm not sure.
Well, for example, in this vaccine study, it was a 6-year study. I don't remember, there were a lot of kids who died in this study. There were a few, like 20% who were hospitalized, and they targeted the study for young kids. I assume that the adults are probably fine.
But that's why the vaccine that was developed is problematic, because it's not helpful for young kids that have never been exposed. Because that's the reason why they are dying. It's because they have never been exposed to the virus. Because of this antibody vaccine.
That's okay. I mean, thank you. The second question is, how far are you to the animal studies with the project?
So dengue is very difficult to do. You need to use immunodeficient mice.
What about the horses that we saw in some slides?
What about them?
The horses we saw in some slides, or other animals?
Horses are even more difficult to work. But, and those are for also viruses, not that. But actually, we are thinking because Zika virus is a much nicer mice model. So because our compound is very good for the Zika, and it's easier model. So we're thinking to do this.
I also have a couple of questions. One of them is more medicinal chemistry. So the compounds you've shown in both cases came from high throughput screening, right? So you get below micromolar activity. So do you consider these as just hits that will then be further developed, hit to lead with medicinal chemistry SAR, so you can increase the potency? Because I think to become a drug, you need to get down to the nanomolar, right? Type of activity. Micromolar seems.
Yeah.
So do you plan to do further SAR and kind of get to a hit to lead type of product?
So
project?
for the entry, so for the those targeted we already did a lot of and the fact that they are covalent inhibitor makes it like a big difference on the other because so the one we have right now is actually quite good. For the entry inhibitor, but it's more like it's more chemistry, it's not production.
And still a medicinal chemistry question now. Do these compounds have like drug-like properties? What's the purpose of that?
So
safety.
Yeah,
Safety.
yeah, I mean, so that's one of those.
That's not medicinal chemistry.
Yeah. No, but like we do like down to that. Yes.
And my second question, you mentioned something interesting. You said that for the second class, for the irreversible inhibitors, that if you pre-incubate the cells, pre-treat the cells, then you don't have viral replication. So would that lead possibly to some prophylactic therapy? Is that the idea? Areas that are highly susceptible?
Well, the problem with the inhibitor is the problem with covalent is like they're washed up with this. You need like a constant supply of this inhibitor. They are very potent for a short period of time. So it doesn't last that long. So that's the problem with covalent. But yeah, it could be. But thank you.
I was just asking, you made that comment, I thought about it.
Yeah.
Because some people have thought about it for malaria.
Yeah.
And before presenting Rafael, I'd like to invite everybody after the talk, networking at 9 p.m. We need to leave the space, but we are going to a pub nearby the Glass House, which is just close to the kayak place here in Canada, just around the corner. So everybody's invited to join us after that for a beer or one. So Rafael Polidoro, he's a postdoctoral fellow at Harvard Medical School, and he studied his undergrad and grad studies in Federal University of Minas Gerais in biochemistry and immunology. And he's going to talk to us about how the gamma delta T cells recognize and eliminate malaria parasites inside the red blood cells.
Thank you. Can you hear me?
Yes.
I will start asking some questions. Zinedine Zidane ou Thierry Henry? Paris Saint-Germain? Lyon? Okay. Au microphone is here. So I'm gonna start to tell you a story of how those very unknown lymphocytes. And lymphocytes are immune cells, so I'm going to introduce everything slowly, but I'm going to give you the final picture right here. They recognize and kill Plasmodium falciparum malaria. And here, as you can probably see but not understand completely what happened before reaching this picture, is like we have blood and malaria infect red blood cells. So you can do smears of blood into a slide, a glass slide, and dry them out. And before doing this blood smear, I place together gamma delta T lymphocytes and their immune cells that we are asking the question, do they recognize malaria or not? And infected red blood cells to assess how they look after this cell culture. And here is how they look in time. 0 hours, they look pretty much like what we would expect a lymphocyte to look like, a lot of DNA and very little cytoplasm. And they go acquiring cytoplasm and acquiring these white dots that will be granules. They use this to kill cells. Whereas the control cells, CD4, very famous because they infect HIV and they cause immune suppression for SIDA, AIDS. They do not respond this way, which makes sense. Red blood cells do not present antigens to conventional lymphocytes like CD4 and CD8. And the parasites, as you can see, the same hours here, they develop a little happy in the first 34 hours, but in the second cycle, every 2 days you have fever, every 2 days you have reinvasion, you have vacuolized lymphocyte red blood cells. So the parasite dies, and nothing happens in the control. They're pretty happy. Those are awesome pictures taken by my wife. So how come like mosquito-borne illnesses, malaria, so individuals live in malaria transmission areas. That's something around this red spot here, and those are mosquitoes. Anopheles gender, and they do. We had 212 million cases of malaria in 2016. It's quite a lot. It's impressive. And every 1 minute and 43 seconds at the time of this slide, one African child died, under 5 years old, died of malaria infection. Right now, was at speed. The vast majority of those deaths are due to the infection with this particular strain called Plasmodium falciparum. You can forget that name if you want, or you can remember. But this Plasmodium causes malaria, and this strain is the killer one. Good news, it's going down. When I started, my first lab was in malaria in 2005, it was every 1 minute. So we're gaining 43 seconds of life for some kids. Bad news, the resistance to drug is rising because like more accessibility, more errors are getting. That's just a natural thing. Malaria is fighting humans since before we were humans. The best strategy to prevent death, infrastructure, and healthcare as other illnesses. Because we have 212 million people infected, they're not all in Africa, but the death happens 90% of them in Africa, 92% precise. And the reason being, it's like people are afraid of going to doctors. They are afraid of foreign medical doctors. And that's the same for Ebola. Like the rate of death in Ebola outside Africa is very low. And sometimes all you need is just really healthcare enough to be kept alive. Like in Brazil, the death rates is really low. Okay, the life cycle. Here again, the mosquito. This was a courtesy of Sabrina. also my wife. Life cycle consists in 2 vectors. And the mosquito goes and bites your skin, and your skin has very good smell sometimes, and some people do not. So don't feel offended if you're not bitten. And you get infected with a sporozoite that come from the meat guts of the parasites, and you can forget all those names, but you have a liver stage that's kind of silent, invisible for your immune system, not totally, but in simple terms. And then you come out as kind of a rosoite, and you go inside the red blood cells, and you're developing ring, just because it looks like a ring. A trove, I would say it looks like a ring too. And schizons that look like a star. And then they go to rosoite and they egress. And we're going to focus in this part. This time here is more or less the time you have fever. So when you have this egress. So every 48 hours, you have fever again. And some of them go, depending on different factors, and nobody knows all of them, they go back to the mosquito in the sexual stage and continue the cycle. Okay, the immune system, simplified. Okay, let's really simplify. So those cells right here that you don't need to remember the name are the COPs, cute COPs. And those cells here are antigen-presenting cells. So they go, so when you have someone robbing someone else, you call the COPs, correct? So they arrive first, your skin has a damage, bacteria arrive, those cells will come and do something, allergic or not. Dendritic cells, and some of the macrophages, and they come from 2 different lineages here, that's a complication we're gonna forget again. They go and see around and take clues. And they present to the SWAT team that there's a flag on front of it. Natural killer cells also can readily kill, but they take like a little bit of time, and just as late. Yeah, T cell lymphocytes, B cells, antibodies, everything that you actually hear about in the media, but they're not doing the really hard work. They come later, they take a while, but they do specific work, correct? So they have villains they fight against. Those guys fight everything. So just to show like they're really real cops, I'm gonna this video. So here we have a macrophage and a bacteria trying to run away. This is the bacteria, and it's really trying to run. Those are red blood cells. So we have here, and this is different. So again, a macrophages. They're nice to study because they stick to glass, so you can people prefer them. But so here's the toxoplasma, and this is a macrophage. And nobody called the cops. So this is just like imagine your cop going home without his weapon or anything. And that's how this was done in the UFMG with a collaborator of my advisor. And you see macrophages are there, not doing much, and then whoop, they actively infect the macrophage. But if you do call the cops before, they're prepared. And that's a cytokine that does that. Okay, remember this name? No! Sorry, technical issue. So this is when the macrophages are activated. You see the membrane is already moving all the time, way more than this one. And macrophage cannot do it. It's actually the other way around. And that's super cool. So just one cytokine activating them, telling them this is something will happen or something happens. They are capable of eliminating pathogens. And this is a video made by my students in Science Clubs Brazil, July. And we did this in 4 days, and they learned, and they did them themselves. They took the video, and they sent it to me. This is Leishmania, tarantula. It infects like reptiles and stuff, so it's not effective for them. This is again a macrophage. And they're going to pay attention. There's a lot of them to increase the chance of happening. So you're going to look at this one right here in the middle, this microphage right here. And see, it twists. Maybe this to grab it, to make a tension and grab it. We don't study much this twist, but the people in physics, they do. Oh, it was automatically playing. Okay. And now I'm going to talk about gamma delta cells that I didn't talk about so far because people ignore them even in the books. They are very complicated, as you just imagine by looking at this. And just forget them. They are the defenders. Okay, so they're not as famous, they're not as cool, but they're badass. So a conventional T cell, so they're like kind of innate. They're not the Justice League. They can fight anything that appears in front of them quickly, and they don't acquire experience in time. So they have very fast response, like in 2-3 days, and then it goes down, but never as down as the level of the lymphocytes that are famous, B and T. And every time they find the, whatever they're fighting for, they will react, and this is response time. Time versus response. Immune response, whatever that means to you. So okay, so we have here a killer, mouse killer cell, and mouse ratinho, little rat. It's much cuter than rats, and they have granzymes that are enzymes that cut things. It's literally like the weapons do. Absolutely shoot you. And granulysin is the authorization from the guy to kill, maybe. And so granulysin open pores and things, and perforin also open pores and things. And so two proteins that make huge holes to make your granzymes go there and cut the entire parasite out, not kill it. But in mice, you have this hole made by perforin, and you have granzyme going, but somehow granzyme cannot go inside the red thing here. And this red thing here is a parasite. Whatever is blue is nucleus. And you see, this is the granzyme protein, not a parasite. See how cool it is for the parasite to escape in mice. But in humans, there'll be a cell that did not reach the red stage. Here, you have controlled infection. And whatever happens is because we have this granulysin, is a protein that mice do not have. Only humans and some other species. That makes pores also in the parasite membrane. So you need both. You need perforin that goes to the mammalian membrane. This is fibroblasts. And then it passes whatever granulysin and granzymes to do the dirty job. And then granulysin makes a hole in the parasite membrane. You have another membrane here. So that's how this happens. Okay, what the fuck are we talking about? So, pardon my French. That's a really bad expression. Let's say that. Malaria, gamma delta inside the toxic granules, perforin, granulysin, granzyme B. This is the only thing you have to remember for this huge introduction. And why this is important, this is a paper from recent paper showing that gamma deltas are required for the induction of steroid immunity during the radiated parasite vaccination. It never worked. Seems that we're kind of finding that they should include some cells they forgot to read about. And the story is very long. So when the receptor was cloned, people found already correlation of the malarian cells in 1991. And they found out that neurozoites primarily stimulate the cells. So when you get the fever, the gamma deltas expand and they become big. So this is size and this is shadow, scatter shadow. And you can see they project more shadow because they become bigger. So that's what this means. Literally what you're seeing here. The small one would be this way, the big one this way. These are actually two cells, but they're both bigger. And they found out granulysin was required in 2004. It's not written, it's written, but you can see. And they found out that the part of the cycle that was important was, again, neurozoites in 2011, 2014, October 2015. This last one showing that actually Scheisen's rupture activates the cell. So they didn't move much in the story. So what is known? Gamma delta react to it, and you have granulizing there. Merozoites are recognized, but they measure indirectly, so they cannot tell gamma delta is recognized and killed, like I did. The granulation happens at distance, is dependent on the pyrophosphate protein. It's actually also important for the mosquito to find you. And killing requires granulizing, but not perforating. That was what was known before. So whatever happens here, you have cytokines, the thing that make the macrophage kill the toxo in the video. They degranulate, and the H2APP is released from the parasites. The gamma delta activate and try to somehow find the infected cells and kill the merozoites. Nobody knows how this thing is bound to the membrane, to the surface of something that's literally just an oxygen machine. What's recognized upon burst? They keep saying merozoites, but they never shown. Granulizing stop growing when, now also don't show, and whatever other things that are not so important. Who are you going to call? A biologist? Me. Who's going to solve all those complicated stuff in a very complicated model of a complicated cell nobody knows anything about? Aventura. The cell culture, and then I start reproducing things from the literature. And yes, PBMC, that's blood cells, your blood cells. They can just, now if I just remove it from you and I test them in malaria, they do respond to malaria. These are the gamma delta, those are control cells, other cell types, B4CD8. This is granulizing, granzyme B and perforin, the 3 proteins that are important. And they do upregulate granulizing a lot. After 7 days, they saw the parasite. They only saw the parasites 1 day. 4 hours, to be honest. Granzyme is not, and perforin is not. Actually, it goes down. And what are those numbers here? Those numbers just show how much is not random. They do not mean anything else than that. But this is not random. The gamma delta T cells are capable of recognizing killing malaria, so it's kind of the same kind of features of before. But here I measured now how much parasites I have infected, and that's called culture I made. And how do you do that? Red blood cells do not have nucleus in humans. Parasites do. They're just stained for nucleus, and you can count how many red blood cells have nucleus. It's super cool. Barrio technique, I wish I could come up with this. So gamma deltas get super happy, and after 1 cycle, 48 hours, nothing changed. Gamma delta did not kill, but after 2 cycles, then you see an effect. More gamma deltas, more Brazilian flag, less gamma delta, less Brazilian flag, and then you see like more gamma deltas, more killing. It's like less parasites I see in the culture. That's somehow cool. You see, the other one was Brazilian flag too. So why they don't kill right away? That was something just, triggering my boss. Picture of this. They need to get to that stage where they're big and full of granules ready to kill. Otherwise, they cannot kill. So all you have to do is give them those 2 things. There are cytokines. Tell them something's happening. That's what happens when you get the first fever. Your body, entire body says, whoa, you have an infection. That's why you have the fever. And when you do that, and you have this high amount of cytokines here, you do degranulate. That's what everybody else measured in the past. Indirectly saying they degranulate means they will kill. Indirectly, you're saying, yeah, they degranulate, you're putting granzyme somewhere, somehow. So they actually, it's true. If you make them big, they degranulate more versus in uninfected red blood cells. So if you do that, activate them into the same experiment, called culture of gamma delta red blood cells, just measuring in the end, well, the impact is not as big as 2 cycles, but also because 2 cycles, they multiply several-fold. But yeah, they actually, more gamma delta, and I should put Brazilian flag here, but it's very complicated in this picture. More gamma delta 1, 3, or 10 against 1 parasite, you have more killing in each stage of the parasite's rings or trophic or Scheisen. So they kill everything, they don't actually care. And if you want a representation of this, is the graph, size versus nucleotide, like DNA and RNA, And you see this is the ring where we have only one nuclei. And more gamma delta you have, this is naturally Brazilian flag. If the software comes this way. It disappears, this rings here. You know, from 8, 1.3, and the control is 11% of infected cells. So it's actually quite remarkable. And do they form conjugates? They form conjugates. So what this graph shows is the number of red blood cell marker, something that every red blood cell has, and anybody else. And this one is delta 2, is a gamma delta receptor. We have a number of gamma deltas here versus red blood cells, and you see that when you have infected cells, it's this gate here, you do have a population that are bound together. And it is a machine called flow cytometer, where you pass one cell at a time, or one conjugate at a time, and you can tell what colors or what markers they have. And this is the uninfected one showing it. Well, this is a like measurement. Almost all of those cells inside those double cells with gamma deltas and red blood cells at the same time have hooks inside that have DNA inside, meaning they preferentially bind to infected and not uninfected. And why this is so important, so if you're passing cells in a machine, 2 cells can pass randomly at the same time. So if it was random, because almost all my cells are known infected, it should be as much here or here. It should be random too, but it's not. You do have a clear preference. And if you want to see that in the machine, like you have a flow cytometer that you pass one at a time, but it takes a picture of it too. And that's what you're going to see here. One infected, one gamma delta, more granular cell, and one red blood cell. And you do see nothing much happens when you look at markers of gamma deltas and CD3 cells. They do not polarize. They are not doing anything with those cells. This is a random cell passing. When it's not random, you're going to see them like this. They're attached to each other, kind of like really binding. And you have the delta 2 receptor that recognizes the red blood cell going towards the direction of the interaction. Here and here. This one is quite cool because it might be happening at that exact time. And this one has already happened because the delta is already going out. C3 is to stick to. And whenever you have purple, you have DNA. So when you don't have DNA, you don't have polarization. When you have DNA, you have polarization. Meaning that they're recognized. Okay, so do the granulizing actually do something, or enzymes? And they do. If you put the 3 proteins together, it's this part of the graph here, you have reduced parasitemia. So if you want to see this is the same kind of graph, you have the first one here where it's uninfected, untreated. And when you have the 3 proteins all together, you have a reduced amount of infected red blood cells. So well, those proteins actually do kill. If you want to see them, you purify infected cells and you have like the granzyme beads, the thing that cuts, labeled with a color, and you look, well, when I put these proteins all together with the red blood cell, do the granzyme B go inside, because that's what actually is going to kill, and you see the red dot only in the ones that have granulizing there, meaning that, and when you measure it, it's quite significant. In rings, it's not, but in trifosilides and in chysins, you have more granzyme B in the presence of granulizing, meaning, yeah, gamma deltas are producing more granulizing and granzyme B in the presence of the parasite, not the granzyme B. And those proteins are capable of killing the parasite. So, I don't know if this one is going to play automatically, but it's just to show the green when you do microscopy of this. So go to the microscope, same colors, and you just look, granzyme B, now I put it in green, red blood cell marker in red, and you see the green is inside the nucleus of the parasite. There's no chance the parasite to come out alive. It's not in the red blood cell only, it's also in the parasite. And granzyme cuts everything you can imagine, so it's gonna kill. So how does granulizing bind to a meta membrane? Because different from the first image I showed, the Trypanosoma cruzi, where granulizing was binding to the parasite, Trypanosoma cruzi, not to the cell, and perforin was binding to the cell. Why? How does granulizing bind to a cell? It shouldn't have this specificity. And the reason probably is cholesterol. Like what determines perforin preference for a cell, a mammalian cell, is the amount of cholesterol it has. So parasites eat the cholesterol. You see, this is the green, cholesterol in green. Uninfected cells have cholesterol around them. And when they are infected, the cholesterol goes to the parasite membrane.
So the red blood cell membrane depletes, reduces the amount of cholesterol and other lipids too. So changes the parasite cause the membrane make them more susceptible. And I reproduce the same result, and you see more or less the same thing. Infected cells where you have nucleus here, you do have less cholesterol in the red blood cell membrane and more in the parasite. And actually, if I just take red blood cells, no parasite at all, red blood cells, deplete the cholesterol using a drug people use themselves to control their own cholesterol, cyclodextrin. And you see the amount of cholesterol really reduces. And the more drug you have, and the more drug you have, or less cholesterol you have, granulizing are capable of lysing. So this axis, the Y one, is lysis of red blood cell. So actually, red blood cells depleted of cholesterol are targeted by both granulizing and actually perforin. Not as much, but also perforin. Okay, another problem solved. Let's just go to solving problems. How this tryphozoite is recognized. So you have this paper showing this butyrophilin 3A1 is a receptor name. Forget the names, just a receptor that presents pyrophosphate, human pyrophosphate, to gamma delta cells when you have cancer. So the metabolism go up so high, the cells show somehow the body saying, whoa, something is wrong, please kill me. And this is the receptor that says that. It's like people tried in other papers I've shown in the literature to show the butyrophilin in red blood cells, and they never shown it. I also did not see. But when you do GHOST, so you hypotonically lyse the red blood cells, so remove the entire inside and let just the membrane there. You probably did this at school if you had some, at least in biology, you do that. So, and you look at the membrane and you go put your proteins through a membrane and you look for an antibody against this receptor, you see the receptor there when you have red blood cell goes from two different patients, and actually gamma deltas have it too, meaning that when you publish a paper saying that gamma deltas can distantly be activated by infected red blood cells, it's just because they can present it themselves. It's not actually recognizing anything in malaria. And my guess is they will kill themselves too. I'm going to jump this result. So when you do something, and I did this as a control for another experiment, I was looking for same thing. Here you have an antibody. The antibody has a color. You pass in the machine and you measure how much of the color is there, meaning more antibody is there or not. That receptor is there or not. Antibodies bind to things. So when you have infected red blood cells and rings and tryphozoite and schizons, you do have a peak of butyrophilin, or this receptor, appearing, and it's not in the uninfected. How can a cell without nucleus, without Golgi, without anything, express something? It was a mystery, but it's not anymore. Butyrophilin, when you look at the MFI of like making the measurement of this, it's pretty significant. Permeabilized red blood cells and tryphozoites and schizons do have this receptor expressed in the surface, and if you treat chemically with things that cut out receptors of membranes, you disappear with the signal here in your trypsin and chymotrypsin. So just by fixing the cell, you make it appear. You don't need a parasite. It might be a conformational thing. That's more or less how we saw it. It's just the parasites alter the membrane so much that the receptor exposes the gamma delta can see, and your cell, red blood cell will be killed. So now the model that I built, it's not published yet. We have a cytokine storm. We have the fever, gamma deltas will get activated, blasts become big, and they deploy via TCR, gamma delta, they recognize infected cells. They deploy granulizing, perforin, granzyme B that can actually kill the parasite. And if this is recognized in any stage of infection, and that will protect you, and I kept saying all this all the time. So I would like to thank my lab and my previous lab, and Jeffrey and Sabrina, my wife, for all the collaboration and the work they did together. They are the malaria specialist, and Judy is the cytotoxic specialist, and gamma delta, nobody was a specialist before, and now I can say I know a little, and my collaborators and the money. Thank you.
question. If memory serves me well, once I read that if you have falciform anemia, you cannot get infected by malaria.
Yes, and you get infected. The malaria is kind of a vortic infection. I mean, there would be more than one reason, but the one I know, and Sabrina can correct me if I'm wrong, is that the membrane tension is so strong that the parasites cannot escape, or sometimes cannot invade. So it's literally, the membrane should have some rigidity, and it has more rigid, so the parasite just cannot get it. But it's literally the shape.
Oh, okay.
Yeah.
So it's not related to the immune.
No. Actually, it's a very bad disease. If you have it and you're not in a malaria endemic area, your life expectancy is lower.
They invade, but the hemoglobin is different in the different cells. Parasites are eating the hemoglobin too much.
They cannot develop
the
it.
hemoglobin. They invade like
I just have a question about, I mean, this is pretty nice that you've been solving problems and answering questions, but I guess the long play is, what is the therapeutic, future for that, especially if you've shown that it seems to be an action at different stages of parasitic cycle.
Yeah.
Which has been the problem, I think, for small molecule drugs, is to be able to hit the parasite at the right time in the cycle.
You're right. So when they shown, I'm gonna say because we have some results already in humans because of the vaccination tests. The paper that shows correlation of gamma delta and vaccines show pre-circulating levels of gamma delta and protection, meaning that you have to have already, for some reason in your life, high amounts of gamma delta when you get vaccinated to make the vaccine work.
Pre-vaccination.
Pre-vaccination. So I would say the way to go is giving some of these pyrophosphates that I was not really talking much about because it's boring, but the things that gamma delta proliferate against, you can give them, it's commercially available, before you give the vaccine. So some days before, 3-4 days, and then you vaccinate the person. That might help, I would say.
Do the gamma deltas get exhausted?
That's about that, yeah.
Can they revert it to like a checkpoint blockade?
No, not that I know, because they do not go for PD-1. They don't go for PD-1 pathway. They upregulate CD16 and become kind of presenting cells, regulatory cells. They do not go exhausted the same way as the cytotoxic cell go. And they actually are so weird because when you look at the blood smears, they look like they make multiple synapses, for example. That should be possible, at least in the mind of the immunologists. You should have one at a time, but I ignore pictures with more than one at a time, but because I can't explain them, and I don't want to go to this with the reviewers. But I think what happens is they are important. Just give a brief introduction of gamma delta that I didn't say. Is gamma deltas are your main T cells when you're born. And they are the majority of your lymphocytes until you are 1 year old or something. So it's when you get malaria the first time. Actually, in some communities in Africa, they do not give names to the kids until they survive the first malaria season. And so gamma deltas might be playing the role of killing them right away when you get like the first cycle. And then they upregulate CD16, that's the antibody receptor. And your B cells will start producing antibodies against malaria naturally, correct? So I think the gamma deltas start killing by other mechanisms after you get your first 48 hours or 2 cycles. The gamma deltas will then kill by opsonization. My guess is we are collaborating with people in Stanford, and they want to assess patient blood. And in patient blood, you can say, oh, it's the first malaria, second malaria, third malaria, so you can see how the gamma deltas will react. It's a very good question.
A quick question. Are there what age groups would gamma delta T cells be more effective? For example, would they be more effective in infants and adults, or
they would be more effective? Yeah,
senior citizens?
It's a good question. It will be more effective in infants. Somehow, it's almost reproducing evolution. The gamma deltas came first too, and they lose importance in time. My guess is because they are innate, they are ready to go, like they are the defenders. They are less important than Batman, but they will be very important at some point, and then you ignore them and you go see Batman. That would be my analogy to saying B cells, T cells will be your main memory and effector cells your entire life, but your childhood until you're 10 years old, it will be gamma delta. And other innate cells that I'm also ignoring.
So, having said that, young adults, let's say people who are over 10 years old, the gamma delta T cells would be not as effective, therefore you have the probability of using higher doses and not achieving the same results as you would in infants, right?
Yes, that's true. If you have to pre-activate them to make vaccine work, you would most likely have to reverse the exhaustion somehow outside the body and replace the same patient.
In the case if they are effective, you have a lesser time period to act. Let's say, for example, perhaps less than 48 hours, right?
Yes and no. And no. They can act that. You're probably not going to die in 48 hours, but sometimes yes. But the mortality is higher in kids too. So it's not super important to be focused too much in adults. Like adults, they can pass repellents and they have drugs and a lot of different things. And one person from Rondônia told me that if you drink one bottle of cachaça, you don't get malaria. So adults can do some stuff.
With so many intercellular interactions, I was wondering what would be the options for monoclonal antibodies? I mean, these cycles of malaria, they remind me a bit the C. difficile infections, and the way they resolve that is with monoclonal antibodies to reduce the severity of the acute phases and just get patients through.
Yes, that was tried. And it's also, I think there's still some clinical trials ongoing with monoclonal antibodies. Unfortunately, malaria is incredibly complex protozoa parasite. So they have a family of proteins that, the same as our immune system, recombine to make antibodies of different specificities. Malaria have different surface antigens. They shift every cycle, a variant scheme. So they literally, you probably have to do 60 different monoclonal antibodies to one patient to make it work.
Yeah, for C. difficile, they have 2 antibodies.
Yeah, I guess malaria is really, we have in our genome like 3-4% of malaria genes. So to see how long this interaction has been going, I guess 8 million years, 10 million years. So, yeah. We are kind of Homo sapiens malaris. And malaria has also some parts of genes, sleeps.
Thank you very much.
Yeah.
Special day.
My wife is French. I learned the accent.
Which is
Perfect city.
Which is why you can
The real
ficha inicial. Informação de se tipo, mais uma lateral.
E eu já preparava. Vocês já vieram para o Miami? Conseguem passar? Eles têm não só o seguro de bagagem, mas de vez em quando eles passam pequenos clichês que eles só pedem seu passaporte, dá uma olhada e passa. Esses, no caso, que falaram no mesmo. É a primeira vez, que é a que você tem lá, depois mais uma vez, depois da mala. Já porque ela abriu a mala duas vezes. Até eu falar assim, porra, eu interessa, e ele fecha a mala. Você já deixa, então eu venho com a camisa de rabo. Nunca barato mais. O cara quer ver o documento, eu posso ver o que passa por cima.
Vou falar isso pra galera do dia. Botar a camisa de rabo. Legal,
Desde 2013.
Você fica muito interessado. Agora no passado da ela é chata.
Você está fazendo exame médico em casa?
E ele faz a fé.
A gente não tem coragem de fazer isso, não.
Não,